chromatin compaction from protein-free DNA to the mitotic chromosome, and the roles of HDACs and HATs in DNA accessibility. Chromosomes are packaged in such a way that they fit inside the cell's nucleus, though they are also organized in a way that they are readily accessible for cell division and replication, as well as for proteins to be read and made from them. A chromosome is a very long molecule of DNA wrapped up by proteins together of which form chromatin. Nucleosomes are an example of organization in the nucleus. They are the basic units of eukaryotic chromosomes. They are made of octamers and DNA wrapped around the octamers. Octamers are 8 histone proteins formed together, 2 of each of the specific histones: H2A, H2B, H3, and H4, which then forms the shape of a spool, which the DNA, around 147 base pairs, wraps around. The DNA then has a strand of linker DNA containing no protein coding information, which leads to the next octamer. This forms a string with beads kind of look to the DNA, and these formations are called nucleosomes. Each octamer also has a tail made of a 5th histone, H1, which causes the coiled structure of the nucleosomes forming the 30 nanometer fiber. The 30 nanometer fiber is then further condensed until the DNA is 10,000 times the starting size, and in the form of a chromosome. The DNA needs to be able to be accessed at any time, which is where the regulation of chromosome structure comes in. There are various ways in which the structure is controlled, starting with how wrapped up it is in localized access to the DNA. Those sequences that are sequestered in the chromatin complexes can be remodeled via the chromatin remodeling complexes, which use energy from ATP hydrolysis to move the nucleosome out of the way in order to unwrap parts of the DNA as well as to wrap new DNA onto the nucleosome at the same time. It is believed that during the process of mitosis that these processes are inactivated to help keep the DNA compact. Another strategy for DNA access is using the H1 histone tails, which are rich in the amino acid lysine, which can be acetylated, neutralizing the tail and loosening the DNA, weakening the stability of the nucleosome. The acetylation of the lysine is done using histone acetyltransferases, or HATs, which allow access to the DNA, while deacetylation can occur using histone deacetylation complexes, HDACs, which tighten the nucleosome, locking the DNA into place, inhibiting access to it. These modifications can affect the stability of the 30 nanometer structures and higher order structures as well because they serve as a histone code, which recruit and activate different enzymes, leading to varying cellular phenomena. These histone modifying enzymes are highly regulated. DNA replication. DNA, being a double-stranded antiparallel molecule, makes it able to be self-replicating due to the base pairing. Each strand can be pulled apart and made into single strands of DNA that can be made into 2 double-stranded pieces of DNA. The use of the 2 single strands of DNA from the original double strand of DNA allows for it to be a semi-conservative replication process. There are origins of replication on DNA where the initiator proteins bind to and pry the 2 strands of DNA apart by breaking the hydrogen bonds using DNA helicase. This process creates the replication bubble formed by the 2 strands of DNA. Due to multiple origins of replication, DNA can be replicated at a faster rate than they would otherwise be if there was only one due to the large amount of DNA contained in a cell. The replication bubbles allow for a replication enzyme to bind to the strands and form the machine, which is a group of many proteins responsible for DNA replication. Single-stranded binding protein keeps the single-stranded DNA from binding back to the other single strand of DNA. The site where the DNA replication begins is called the replication fork, a Y-shaped junction where the old DNA is separated and new DNA is synthesized with the old. The replication forks forms on either side of the bubble, moving in opposite directions. This process is called bidirectional. The DNA is synthesized at the replication fork, 5' to 3', which is fine for the leading strand because it can be synthesized continuously, while the lagging strand is made discontinuously. The lagging strand synthesis of DNA is made up of Okazaki fragments, which are later joined together, forming the one continuous strand of DNA. This is why the replication fork is considered asymmetrical. The start of this machine that produces the new strand is called DNA polymerase, which uses the old strand as a template to add the new strand to, forming phosphodiester bonds between 5' and 3' ends of the DNA. The nucleotides enter the reaction site of the DNA polymerase as deoxynucleotide triphosphates, DNTPs, DGTPs, DCTPs, DTTPs, and DATPs, which is what gives the energy needed to power DNA replication. To keep the rate of replication in the process high, a clamp protein is placed around the DNA using a clamp loader as a ring and interacts with the DNA polymerase to keep it from floating away each time it adds a nucleotide. DNA polymerase is self-proofing, meaning it checks itself as it is adding nucleotides to the DNA through the feel of the DNA. Due to the base pairing of AT and CG, the DNA strand is considered smooth, so when the wrong base is added, the DNA is no longer smooth, signaling to the protein that something is wrong and must be fixed. The DNA is fixed by taking the wrong nucleotide off by cutting the phosphodiester bond and restarting the process with the correct nucleotide. DNA polymerase is unable to begin synthesizing new DNA de novo or from scratch. It needs DNA polymerase to begin the process because it is able to produce strands of complementary to DNA de novo. The new strand of DNA then is started with an RNA primer made by an RNA polymerase called primase, which is only about 10 bases long. This provides the 3' end for DNA polymerase to begin adding new nucleotides to. The leading strand only needs one RNA primer because it is synthesized continuously, but the lagging strand needs many primers because it is synthesized in the segments, Okazaki fragments, so each fragment has its own primer. The fragments are joined using 3 enzymes. The first enzyme breaks up the RNA primer on the fragment, while the second, repair polymerase, places new DNA in place of the RNA, and the third enzyme, DNA ligase, joins the sugar phosphate backbones of the DNA together. Primase is unable to place RNA primers under itself. It is only able to place primers in front of itself, which leaves areas of chromosome unable to be replicated, which, if continued and allowed to repeat, would shorten chromosomes each time it replicated. Telomeres have been evolved to allow for DNA replication machinery to land on the DNA, eliminating the shortening effect. Telomerase is attracted to the ends of the chromosome, which is the enzyme that makes telomeres. These enzymes have their own RNA template strands, which allow for the creation of the shortened DNA sequences. All of these things in this process of semi-conservative replication are occurring at the same time and work together to form new replicated pieces of DNA on both strands. DNA mismatch repair and the 2 mechanisms for repairing double-strand breaks. One of the repair mechanisms of DNA is the DNA mismatch repair system, which replaces the bases that are paired wrong in the DNA. This system recognizes the mutation in the same way that DNA polymerase does by the distortion of the DNA, smooth versus not smooth. The mismatch is cut out of the DNA and the old strand is used as a template to fix the error that is there. Bacteria distinguish these new and old strands of DNA through methylation of the old strand, but it is not known how eukaryotes decipher what strand is new and which is old. This repair system is our primary defense against somatic mutations and cancer, though if there is a mutation in the system, it can cause inherited predispositions to occur. Another form of mutation that occurs is double-strand breaks, which is where DNA is completely severed into 2 separate DNA molecules, which is a potentially life-ending mutation. There is no intact strand in this case to use as a template to fix the DNA with, meaning the above-mentioned repair mechanisms cannot be used. One of the ways that this can be fixed is non-homologous recombination, which takes the two broken pieces of DNA, cuts them off, makes them so they can bind again, and uses DNA ligase to place them back together, which can lead to the loss of nucleotides. This would be fine if the area of DNA is non-vital, but if the area is vital, it can lead to total loss of function in the cell. Another form of repair to double-strand breaks is homologous repair, which is where the information from a nearby similar strand of DNA is crossed with the broken strand to serve as a template for repair. One of the broken strands is chewed back, allowing for the crossing of an intact molecule. This is called strand invasion. Where this occurred forms a branch point where the two strands, one from each of the molecules, crosses, allowing for repair of the invading strand using repair polymerase to elongate it and synthesize new DNA. This process is completed by nicking the invading strands, returning them to their original positions on the strand, and welding them in place using DNA ligase. This process is the most versatile form of repair due to the ability to get rid of mutated DNA and due to the use of it during prophase 1, allowing for higher genetic diversity. The assembly and purpose of the pre-initiation complex. The pre-initiation complex is what tells RNA polymerase II that it is able to initiate transcription. It requires general transcription factors that assemble on the promoter and position the RNA polymerase on the exposed template DNA strand. The process begins by the GTF, TF2D, binding to a short segment of the DNA composed of T's and A's, the Tau box. This binding creates a landmark for the rest of the GTFs to bind and position RNA polymerase II. The TF2A, TF2B bind to form more of the pre-initiation complex before TF2F and TF2E bring the RNA polymerase to the strand and position it before TF2H places a phosphate tail onto the RNA polymerase II, which is the final piece of the pre-initiation complex. Once this process has been completed and transcription has begun, the GTFs are able to dissociate and initiate another round of transcription. Post-transcriptional mRNA processing. The first step to the process of mRNA is by adding the 5' cap, which is a base attached atypically, marking the start of a processed mRNA. The next step is the addition of a poly-A tail to the 3' end, which is about 100 adenine bases in a chain using multiple enzymes, marking the end of the correct mRNA. These additions also make the mRNA more stable when leaving the nucleus for circulation and sends out a signal letting the cell know it is ready for translation. These mRNA sequences must be edited, though, to remove the non-coding sequences they contain. Introns. Introns are considered remnants of extinct viral infections, magnets for mutation, and other hypotheses for what they do, while the coding sequences that they contain are called exons. The splicing of these introns occur using a spliceosome, which looks for the limited conserved sequences at the ends and in the middle of the introns. Before ligating the mRNA exons back together, the 5' end that is freed in this process is attached to an adenine in the middle, making what is called a laureate, which saves NTPs for other processes. There is also the alternative splicing method that allows for the option to cut out exons or leave them, giving the ability to produce many proteins with different sequences, shapes, and functions from a single gene. This allows for an increased protein potential. Translation. mRNA is coded as nucleotides, but then they are taken from 4 bases to 20 amino acids, which are coded using 3 bases in a row. The 3 DNA-RNA nucleotides are called triplets or codons. These are part of the genetic code, the dictionary for bases to amino acids. There are 64 triplets possible to be made from combinations of the 4 bases. This means that more than one codon can code for each amino acid, which helps prevent mutations from affecting the protein because a mutated codon could still code for the protein needed at the time. These codons are read through reading frames, which there are 3 possible ways to read. The first is 1. Open reading frame starts with a start codon, AUG, and ends with a stop codon, or nonsense codons. This is also considered the correct reading frame because it looks for the start codon to read from and stops reading when it reaches the stop codon at the end of the reading frame. The codons that make up the mRNA strands are not able to bind to the amino acids that correspond to them directly. Instead, they use molecules that are adapters to bind. These adapters are called tRNAs, which have anticodons on one end, which are able to bind directly with the amino acid on the end. The tRNA is the transfer RNA that takes the amino acid and moves it to the ribosome, though it also serves as the translator RNA from nucleotide to amino acid because it is the only molecule that speaks both languages. tRNAs are also a self-base pairing molecule. tRNAs have 4 extensive intramolecular base pairing regions that form a cloverleaf structure, which then folds in half to form a more L-shaped structure. In this configuration, the tRNAs, anticodon, and amino acid attachment are still readily accessible. The base pairing abilities also allowing for a form of pairing called wobble, which is where the single tRNA recognizes multiple codons, usually in the 3 area of the codon. There are 31 tRNAs that allow for some to only code for one, but most wobble, meaning they bind with multiple codons. Before the tRNAs can bind to a specific amino acid, the tRNAs get charged, meaning an enzyme called aminoacyl tRNA synthetases links specific bases with corresponding amino acids. Ribosomes are the complexes formed of the RNA and protein molecules to synthesize proteins. There are subunits that are responsible for the different steps to translation. The 2 subunits come together with the mRNA in the middle of it to facilitate this process. The smaller subunit matches mRNA codons to the tRNA, allowing for the codon recognition needed to form the proper tRNA and amino acid strands. The larger subunit of the ribosome forms the peptide bond, the bond that links the individual amino acids together in a chain. The larger subunit, in the larger subunit, there are 3 areas or sites that the tRNA must travel through. The first site is the A site, aminoacyl tRNA. The second is the P site, peptidyl tRNA. And the third site is the E site, or the exit site. Each site has a specific process that it must perform in order to form the protein molecule. The 2 subunits come together with the mRNA in the middle of it to facilitate this process. The process starts with the 5' end of the mRNA getting surrounded by the pieces of ribosome that are assembled. To initiate the process of translation, an initiator tRNA associates itself with the A site of the ribosome, allowing it to bind with the 5' end of the mRNA in the middle. This initiator tRNA scans for the start codon, AUG, with the anticodon to it, CAU. When this is found, the initiator binds to it, allowing for the pair to sit in the P site with the larger subunit closing the ribosome. This process establishes the reading frame of the mRNA. In eukaryotic cells, the initiation factors, EIFs, bind to the 5' end of the mRNA and the small ribosomal subunit, which allows for the recruitment of all the pieces needed to perform translation correctly. Without the 5' cap that was added onto the mRNA, there would be no initiation or recruitment of all the pieces of translation, which shows how important that step was. The tRNA then, so long as it has the correct anticodon and the correct amino acid bases, enter the A site of the ribosome, which binds codons to the anticodons in the correct order via base pairing. Then the amino acid chain binds to the amino acid of the tRNA in the P site and transferring to that tRNA, which allows the ribosome to shift to the next codon and moving the tRNA that was in the P site to the E site in a process called translocation. This also moves a new tRNA amino acid pair into the P site, moving down the tRNA, forming a protein. Only 2 tRNAs can be in the ribosome at the time, so when the ribosome shifts, taking in another tRNA, the one in the E site is moved out of the ribosome. The mRNA is moved through the middle, being translated into the tRNA, and amino acids are bound to it, leaving the ribosome as a tRNA and amino acid chain. This is continued until the stop codon is reached and the process is terminated. The termination is easier to understand as the stop codon signals that is the end of the gene and is positioned under the A site of the ribosome, which signals the release factors to catalyze the process of releasing the polypeptide amino acid chain from the tRNA that is located in the P site, which also causes the dissociation of the ribosome and the mRNA that is let go from the middle. The protein that is formed from the amino acid chain into the proper protein shape goes to perform the function that it is meant to. Some RNAs are able to form into a shape which gives the strand a function, and because all protein machines are called enzymes, and the ribosome is 2/3 RNA, which gives it the function to form proteins, it is called a ribozyme. This is the RNA world hypothesis, which shows that RNA can have a function. In order to keep the protein output of a cell high, there is a formation of a structure called a polysome, an mRNA with multiple ribosomes on it, translating at the same time. When the first ribosome moves off the 5' end of the mRNA, a new one binds onto it, which results in a higher output of protein because more than one is being synthesized at the same time. Antibiotics are considered protein inhibitors of prokaryotes as it prevents protein synthesis through translation, and no proteins means that you die, which is also where antibiotic resistance begins because it is a learned defense mechanism to allow proteins to continue being made. Proteases are the enzymes that destroy proteins when they are no longer needed, they are misregulated, or are causing destruction. When the protease destroys the protein, the amino acids that were used to make it are recycled for new protein formation. The most used protease is a proteasome, which shreds the protein. The proteins meant for destruction are clearly marked with a specific and unique tag. Combinatorial control and mediator. Regulatory genes are thus spread across the chromosome with DNA between them that can be unrelated to the target gene. This is important because the genes are regulated by a committee of regulatory proteins. This committee allows for the genes to be responsive to multiple signals, allowing for combinatorial control, a group of proteins working together to control the expression of a gene. Each gene in the eukaryotic cells are controlled by multiple gene regulatory proteins, repressors, and activators, and each one is normally sending its own signal to the cell. The resultant gene expression is generally the committee decision, which takes what each protein signal says and sees which way the scale goes. The many transcription factors that bind to the DNA and loop out the sequences that they don't need to reach the target gene promoter relays the signal saying to start transcription or not. Activators in eukaryotes aid in the initiation of transcription by allowing RNA polymerase II to leave the promoter, meaning they do not affect the recruitment of RNA polymerase II to the promoter in the first place. The mediator is a protein complex of about 20 proteins that sits on the RNA polymerase II and passes the signals that it hears from the activators and repressors of the gene. The mediator sends the first signal to the RNA polymerase II saying whether or not it can proceed with transcription. This is also what allows for coordinated regulation because the same transcription factor is used to bind to the site and turns the genes on or off at the same time, meaning that every gene needed for a process is turned on and off, keeping them working together. RNA interference. RNA interference is a mechanism that leads to the silencing of genes, which requires the formation of double-stranded RNA. Double-stranded RNA is recognized by the RNA interference in all of the double-stranded DNA, and single-stranded RNA with the same sequence is destroyed. The protein that is coded by that sequence is never to be made again because the gene for it is gone permanently. Any mRNA that may have been made from that gene is also marked and destroyed immediately. RNA interference is thought to be evolved for defense against viral infections, which is the primary source of double-stranded RNA compared to most RNA in living cells that is single-stranded. RNA interference is triggered by double-stranded RNA that is base paired, 21 to 23 base pairs long. Those that are longer are cut by a dicer, an enzyme. These are called small interfering RNAs, or siRNAs. These siRNA fragments are then bound to an RNA-induced silencing complex, or RISC, which uses one of the two strands that it is bound to on the siRNA to identify other RNA molecules with the same or complementary sequence. Once the sequence is recognized by the RISC destroys the mRNA. The mRNA that is destroyed by the RISC leaves two unstable fragments that are used by an RNA-dependent RNA polymerase, RD-RP, to make the double-stranded RNA, which does not require a primer. The new double-stranded RNA is then cut by a dicer, which gives more siRNA bound to RISC, which starts the process over again. Though we lack the ability to amplify RNA interference because we do not have RD-RP, roughly 50 of these siRNAs can silence thousands of mRNA and is one of the most potent forms of regulation known in eukaryotes. In laboratory situations, RNA interference is used as a way to shut off target proteins using artificial genes that form a loop, creating double-stranded RNA when transcribed, or a single region with convergent promoters, which also forms double-stranded RNA when transcribed, allowing for specialized treatment.